this is just for the students that currently I am an assistant professor in plant biotechnology at Gujarat Biotechnology University. That's my email ID for those of you who would like to be in touch regarding research and academic advice or anything related to your area and interest. And I basically work in three broad areas. I am working in the plant pathology area and plant microbe interactions, and I'm also interested in microbial diversity and bioprospection for beneficial metabolites that can be used in agriculture applications. So, with that, I'll take you to a ride of plant fungal interactions. So, this is already rainy season in almost all parts of India, and you all must be experiencing fungi here or there. And sometimes they are pleasant to watch at in form of mushrooms, and sometimes they are really ugly to look at in form of diseases. So my focus being a plant pathologist is more towards the plant diseases, and I'll introduce you slowly to the concepts. So I'm very fascinated by mycelia spores and research. So that's what I do and that's what I love. So I'm a mycologist. Mycology is a study of fungi. And I'm also a plant pathologist. That means I study diseases in plants, particularly caused by fungal pathogens. So how
सतीश जी
interesting
कैमरा बंद कर लीजिए. सतीश. Yes.
So, how
Please continue, madam.
interesting these interactions could be. So, there are multiple complex interactions happening with between microbes and the plant. In the ecology, they have some significance or relevance. We don't know. So, we'll figure it out in my as I progress in my talk. So, like human body is full of microorganisms. We are always reading about gut microbiome, liver microbiome, kidney microbiome, and how they are important for the overall survival of the human beings, how they are beneficial, and how they regulate overall human physiology. So, in the same way, plant also has their own microbiome. This can be present in above ground tissues or the underground tissues such as root. And these interactions are also complex and could be beneficial, detrimental, and playing a complex network of events, biochemical events, and regulating plant physiology. So this could be good and bad. So in the beneficial way, these microbes exist as endophytes or epiphytes. So endophytes are the organisms which are present within the plant tissues, and epiphytes are the organisms which are present on the surface of the plant parts. So these endophytes or epiphytes form kind of a symbiotic relationship with the plant, host plant, and help in the growth, promotion, phytohormone secretion, and all kind of metabolic activities, nutrient acquisition, and etc. While in the detrimental side, they could be pathogenic and causing severe diseases, yield loss, stunted growth, and overall decrease in plant health. So the beneficial side could also be regulated by different rhizobacteria, which is a very common term that students generally know. The nitrogen fixing bacteria that are present in legumes, and the other soil microbiome that are regulating plant growth. In return, the plant is providing shelter to the plant, to the these beneficial organism, and also sometimes they provide carbon source in form of the food. So again, as I mentioned, there are epiphytes, endophytes, and pathogens, and there are all kind of complex interactions happening: secretion of enzymes, secretion of phytohormones. The root plant is also secreting signals to attract these pathogens. So all these complex interactions are occurring on a daily basis, and they are supporting the overall growth and physiology of plant. So coming to fungi, so fungi is my organism of interest. So in microbes, I work with fungi, and fungi can be your friend or foe. How they can be friend? They are very popular decomposers, and they help in recycling of organic matter. They also produce different industrial enzymes and antimicrobials, And most of you must have heard about mycorrhiza. So mycorrhiza is again a beneficial interaction of fungi with plants, and they also act as biocontrol agent. A very common and popular example is trichoderma, which is being widely used in by in nurseries and in small home gardens as well as in fields. Four is when these fungi start causing some kind of detrimental effects on either the consumer, which is human, or to the plant, which is its host. And plant diseases are a common example. You can, especially in this season, wherever you look around, you can find one or the other mold growing. And you must have noticed that the food also gets spoiled easily because fungi loves moist and humid environment. In addition to the diseases, fungi also are notoriously producing several mycotoxins which cause allergic responses in human beings. So, if you consume some grain or some fruit or some food product which is contaminated by fungi, uh, which secretes some kind of mycotoxins, it can cause detrimental effects on your health, and in severe cases, it can also lead to neurodegenerative disorders. So coming to fungi, so fungi, this picture is again for the students because visually they can more relate to what all I've been talking about. So fungi, popular, everybody relates mushroom to fungi. So mushroom are widely used in medicine as medicinal purpose, also as food. We love mushrooms, and they also produce several industrial enzymes like penicillin, which is a very common enzyme, and very common antibiotic which is widely used. And in food industry, fungi are used as yeast to produce fermented products like bread, cheese, wine, etc. And in plant they help as forming symbiotic association as mycorrhiza with algae. They form symbiotic association known as lichens. And that's the beneficial side, and these are the different pictures of plant disease symptoms caused by fungi. And also in humans, candidiasis and oral thrush and nail infections, which are common diseases caused by fungi. So there are diverse ecological roles and importance of fungi. And coming to the plant interactions of these fungi, so they also are multidimensional. They can cause benefit, also they can cause harm. So benefit is obviously in symbiosis or as endophytes, as mycorrhiza, where they are benefiting the plant by providing nutrition, by providing protection, by providing biocontrol properties. And the harm is in parasitism, where they actually lead to the destruction of the crop. So that is something which is of very importance because India is an agriculture based country, and we cannot because of the rising population, we also have global demand for food supply, food security, and crop loss is something which we don't want. So this animation again is of mycorrhiza, and how these mycorrhiza, the purple part shows the fungal network which is associated with the plant root, which is shown in yellow. And this, just visually you can see how much surface area of the root is increased and how much it is helping the plant to acquire nutrition directly from the soil. This could be water, minerals. So that's the benefit that the plant gets when it has a healthy association with the fungi. Now, pathogens, coming to fungal pathogens, they can be of three different categories. So, one is biotroph, one is hemibiotroph, and the last one is necrotroph. Biotroph is a fungi that is always growing inside plant tissue without causing much harm because its survival is entirely dependent on the plant. If the plant is dying, that fungi will not survive. So it never leads to the death of the plant, but in turn, it affects the overall productivity. The common example, just to explain you further, could be rust or powdery mildew disease, where they cause visible lesions in the plant leaves or aerial parts of the plant, but the plant is still not dying. But overall, it is acquiring nutrition from the plant, so that's why plant doesn't have much enough resources to allocate to the pods and fruits and the other byproducts. So that is something which is of concern with respect to biotrophs. Coming to hemibiotrophs, they have a like dual cycle. They can survive as a biotroph in the initial phase as long as they are comfortably surviving. As soon as the situation become adverse, they feel some kind of stress, they switch their cycle to disease one. So that this switch is the pathogens that cause this have this kind of switch are called as hemibiotrophs. And the third category is necrotroph, which are directly causing diseases in the plant. The common example is botrytis. So this is again visually how biotrophs are living inside plant tissues, and they are colonizing in the intracellular spaces, also penetrating inside cells, but they don't cause any disease. The disease is shown in brown, and in case of hemibiotroph, they start off as biotroph, but when the situation becomes unfavorable for the fungi, they switch gear to the disease mode. And necrotroph, as in the beginning itself, they start causing disease. So we all agree that plant health is important. However, fungal and other plant diseases caused by different pests contribute to overall about 40% of crop loss each year. That's a huge loss in crop yield, because if 40, just imagine if 40% is lost, how can you feed the entire earth population with the remaining 60%? And a lot is also lost as byproducts, like the entire crop yield, pods and everything also goes to agriculture waste. So we are ultimately left with around 50% or 40% to feed the globe. So that's why it's very important to take care of plant diseases to address this issue, and how we can safeguard the entire humanity from dying from hunger. And also, as advocated in the Sustainable Development Goals, there are four goals which are entirely focused on safeguarding agriculture and our forests. So there is zero hunger mandate, there is good health and well-being mandate, there is mandate for climate action and life on land. So all these to promote agriculture, tackle climate change, and to achieve a sustainable development. So as long as like there was civilization, they have been growing crops, because in the beginning they were hunting, and as soon as they learned about tools, they started agriculture. So in the Asian civilization, there was not much awareness about diseases, and whenever there were the field was infected, they used to blame God. So that was very easy for them to just blame God, that okay, God is angry with me, we need to give some sacrifice or offering to the God, and then only it will, because the scientific knowledge was not there. So they have battled, like as long as there was civilization, they were growing plants, they are battling with disease. But only now, or with the advent of scientific discoveries, we understand that these are not act of God, they are microbes actually behind that. So, how do diseases occur? Diseases occur, there are three factors associated, and it's a interplay of these three factors. So, even if one of the factor is not favorable, the disease will not occur. So, what should be there? There should be a pathogen, first of all. So, if there is no pathogen, obviously disease won't occur. So, there should be a pathogen like fungi, bacteria, or a virus in case of plants, and there should be susceptible host. So, if the host is resistant to the pathogen, disease will not occur again. So, the host has to be susceptible for a disease to be formed. And then the conditions, the environment should be favorable. Again, if the environment is not favorable for the pathogen, for example, if it's too dry, too much temperature, it's difficult for the pathogen to survive as well. So it's an interplay of these three factors, then only a plant disease will occur. So these are some of the important research questions which I'm trying to address in my like past and current research. So I'm interested to know what is the causative organism, like which pathogen is causing the disease. So I have my colleagues who bring me some sample from their home garden, or if from the sampling, if they went for to collect their sample, they also bring me samples for diseases. So first and foremost thing for a plant pathologist is the accurate identification of a plant pathogen. If you don't know the cause, you can't do the treatment. So that's why diagnostic is very important. And then to understand the mechanism, how these pathogen infect the host, and what all these we as a researcher can contribute to prevent plant diseases. And recently there's a lot of multiomics approaches that are relevant. There's a lot of data lying in public databases, GenBank, NCBI, that can be used. We can mine proteins, we can try to find information relevant to plant pathogen interaction studies. So, a bit background about plant immunity. So, like in humans, we have adaptive and innate immunity. So, there is innate immunity, which is present by birth, and there is adaptive immunity, which we acquire by exposure to pathogens. However, in case of plant, there is no adaptive immunity. Plant responses are only innate. They are defined genes and already present in the plant, and there is nothing like adaptive immunity. There's especially no immune cells or no antibodies in plants, and whatever response is there, it could be local as well as systemic. So coming to plant microbe interactions, um, um, so just like humans, we have skin barrier, which is the first line of defense. So in case of plants, the first line of defense is hairy growth on the leaves, waxy cuticles. There are in some plants with thorns which protect them from insect pests. So these are our first line of defense. Like we say, first line of defense in humans. In plants also there is some bit of innate these defense protection layering present. But this is not enough because pathogens are constantly evolving. So plant also need more. Um, so it's a constant battle of survival, and slowly and steadily, plants are also evolving genetically to ward off these pathogens. And there are three lines of plant immunity. One is PTI, pattern triggered immunity. One is ETS, effector triggered susceptibility, and the third is effector triggered immunity. So, coming to a brief introduction about PTI. So, PTI is basically triggered by molecular patterns present on the pathogen surface. So, it could be in case of bacteria, the flagellin, or or some effectors. Effectors I'll explain in detail. These proteins that cause diseases, virulence factors, and there could be other proteins that are present on the surface. So these are pattern associated molecular patterns which are recognized by plant receptors known as PRRs, and they trigger downstream signaling events known as pattern triggered immunity. So these are innate responses. And then the second one is effectors. So effectors are small virulence proteins that are produced by the pathogen. They are delivered inside the plant cells, and they hijack plant defense response and cause infection. So in that case, it's effector triggered susceptibility. Susceptibility means disease is happening. But in certain case, the plant also has certain resistance proteins which can recognize these effectors and cause effector triggered immunity. So these are three different immune responses broadly that are happening in the plant. And a bit about the multi-omics approaches. So we all know that from DNA, genetic information, the whole genome sequence, we can get the information about genes present in a organism or a pathogen. From transcriptome, we can analyze mRNA level transcription expression data. From proteomics, we can analyze all the functional proteins present in a pathogen or plant. And using metabolomics, we can understand about all the metabolites that are present in the plant system or the pathogen system. So again, All these approaches can be included in plant pathogen interaction, and we can do all kind of SNP genotyping, QTLs, and we can identify resistance genes in the host plant, and we can find targets for breeding, molecular breeding approaches. And from the pathogen side, we can identify pathogen effector candidates and host specificity genes. So now I'll switch gears from introduction background to the research data. So I'm talking about just one story which I did during my postdoc as a national postdoctoral fellow at Regional Center for Biotechnology in Faridabad. My mentor was Dr. Divya Chandran. She is an associate scientist there. So in that lab, our crop was pea, and pea is a legume crop, and it's very important nutritionally as well as biologically. It helps in improving soil fertility by fixing biological nitrogen fixation, and its roots architecture also improves soil quality by providing organic matter structure and porosity. This straw of the crop can be used as cattle feed, and the food itself, the pods, pea pods, and peas are vegetarian superfood. They are rich in fiber, proteins, vitamins, and minerals. So, how the disease is happening here? So, I was working on powdery mildew disease, which is an obligate biotroph. So, as I already mentioned, biotroph are always requiring a living plant tissue to survive inside the plant, and they derive nutrition from the plant. So, how they cause infection? The spore of the fungi will land on the surface of plant tissue, aerial part, and they will form a germination pore. This germination pore will form a penetration peg and eventually form a specialized structure known as hostorium. Hostorium, in case of biotroph, is the site which synthesizes effector molecules. Effector molecules are small virulence proteins formed by the pathogen that hijacks plant's defense machinery. And this is also the site which helps in the acquisition of nutrients and water molecules from the plant tissue. So it is also the feeding organ as well as producer of effectors. There's another class of effector which are also secreted by the epidysorium and the initial formation during the formation of penetration peg. And this is how Hostoria looks like under the microscope. So these are my own images. I isolated Hostoria from the infected pea leaf. And my approach was to do these RNA sequencing of the isolated Hostoria because I was interested to understand how this tiny cell, the modified cell structure that is formed during infection by the biotrophic fungi, how it is able to do so much, cause so much trouble to the pea plant. So we isolated Hostoria, did RNA isolation followed by transcriptome sequencing, and all this data was processed bioinformatically for the prediction of secreted proteins, which is known as secretome. So these are common tools: Signal P, Target P, TMHMM, Big Pi. All these were used. That is a standard pipeline for prediction of secreted proteins, and we identified around 622 proteins which were secreted. So that's how the symptoms look like on the crop. They look like white powdery appearance, and it can be present on the pea pods as well as on surface of pea. And they also infect model plant Medicago truncatula, which is model legume plant. So the Hostoria was isolated and sequenced, and around these effectors were further analyzed bioinformatically for the presence of conserved protein domains, and the diversity was also analyzed in this plot. You can see the center shows the diversity. So around 13 different protein clusters, effector clusters were identified, and they all had conserved YFWXC domain, which is a characteristics of powdery mildew effector proteins. And further, their expression was checked via qRT-PCR. So the black line shows the expression in Hostoria versus the magenta lines that show the expression of the effector in the epiphytic mycelia, epiphytic which is on the surface. So as it was apparent and very obvious that all majority of the effector are highly overexpressed in the Hostoria, which is the key site of the synthesis of effector proteins. Further, we also did a time course to analyze the time specific expression of these proteins, selected proteins. We didn't do all the 167. We had a subset which was of more interest to us. And we also observed that between 12 to 48 hours is a specific time of the formation of Hostoria inside the host plant. And these effector candidates were typically overexpressed during the 12 to 24-hour window. Then to validate them functionally that yes they are having some role in the disease, so we did a transient silencing. We used double stranded silencing approach, and these graphs show that we took three candidates, CSCP 1, 9 and 83, and as compared to control black bar you can see that these three are uh, effectively silenced. The expression was effectively silenced. And also, visually, you can see that as compared to GFP control, the silenced double-stranded silence effector protein controls were having visibly less symptoms. We also checked them under the microscope, and the same result was observed that there was reduction in the virulence. So, upon silencing, there is reduction in virulence. That means these are having role in infection or the disease of the candidate pathogen. So pathogen name I have not mentioned. Pathogen was Erisifi PC. Further, we also checked the subcellular localization of these effector proteins, and CACB1 localized in the nucleus, 9 localized on the cell membrane as well as the nucleus, and 83 again localized in the nucleus. We and in case of powdery mildew, there is a class of effector proteins known as RALF uh, effectors. They are RNAs like effector proteins. They have some kind of uh, ribocatalytic activity, and our candidates we check them with the known RALF effector. So PEC 1054 is a known RALF effector, and they all had a structural resemblance and possess RNAs like domain in their structure. So this was a cover article in the journal Molecular Plant Pathology, and from this work we got two publications, and really it was good amount of work that we could achieve in 2 years of time. I'm really thankful to my group there, and especially to ma'am for all the support. So with that I would like to give a break, and I just like to reiterate that fungi have a very important and ubiquitous role in our environment. They could be a friend, they could be foe. It's up to like us how to not up to us, but it's for the researchers to take the ship from foe to friends. And like I explained in my research, these effective candidates were identified. Now the next step could be to identify the plant proteins that were being targeted by the pathogen, and that's how we can maybe contribute to some crop improvement strategy by identifying the susceptibility factors that can be further used for targeted genome editing for crop improvement. And also we would I would like to emphasize how the multiomics approaches can help to speed up the research. It could be applied to any area of research and can have like immense amount of information can be generated by just one technique. So then we can further functionally validate everything and then contribute to the scientific update. And for the students, especially in the areas of plant fungal interaction, there is a lot of things that are yet to be explored. Not a lot of our genes, resistance genes, are known from plants. Host specificity factors still need to be, there are a lot more work to be done. and we can also determine the virulence mechanism of pathogens. So we need to dive into the area of plant fungal interaction and multiomics to kind of get more information, and then we can use that information for crop improvement strategies. And with that, I would like to thank all my funding agencies that have supported my research throughout. First and foremost, for my PhD, I thank CSIR. I have not included all the work because I wanted the students to be aware with one topic. Um, I hope I could do that. I could do the job. And the next is thanks to special thanks to SERB for the NPDF fellowship, with which I could all the work that I've demonstrated was under that fellowship program, and also to my postdoc supervisor, Dr. Divya Chandran, and my PhD and postdoc mentors, Dr. Shunoy, Dr. Pinaka, Dr. Stanley Freeman, and Ms. Marcel Memo. And uh, currently, I am working at Gujarat. I'm working in the same direction. I'm trying to identify virulence factors associated with anthracnose disease. So I've been supported by DST Gujarat for the research funding. So I also like to thank my workplace and current funding. So thank you all for your attention, and I'm